what are potential options here? So what explains the symptoms? Or what has led to the rise of C. diff infection here? All right, we'll move on then. Think about that one. Maybe an essay question on the exam. What researchers are thinking of doing nowadays, though? Using fecal transplants. So the idea of a fecal transplant is not to necessarily treat the infection directly with antibiotics, but rather to treat the infection by restoring the natural microbiome. A fecal transplant is not like an organ transplant or a blood transplant. All you have to do is match somebody who has a similar diet as yourself. Usually you're looking for a close family member who's not afraid to poop into a cup and then have that go into your stomach somehow. The ways that it's being done right now, most often, it's through the nose, straight down into the stomach, and then into the gastrointestinal tract. Though some companies are playing with alternatives, such as using sporulated, freeze-dried bacterial spores that would then undergo replication once they reach the gastrointestinal tract. And this is just some data from from an article in 2013 in the New England Journal of Medicine. They were looking at percentages of individuals who were cured from a C. diff infection without relapse of a second C. diff infection after receiving donor feces. And then the third bar and the fourth bars there represent after being treated with vancomycin and after being treated with vancomycin and a bowel lavage, which is basically taking like what's inside of you and rinsing out the inside of your gastrointestinal tract. So trying to remove the Clostridium by flushing out the system. And what they find in these basic results is that one time through, 80% of individuals are cured with a feces transplant. Overall, 94% are cured by a feces transplant, either one or two or three of them. And then with vancomycin alone, only 30% of individuals have no relapse in infection. Vancomycin is a broad spectrum, we consider to be a relatively broad spectrum antibiotic. And it is going to not only kill Clostridium, but it's also going to continue to deplete the natural microbiome, which is in the gastrointestinal tract. So the argument here then is that this doesn't really do anything beneficial. More often than not, it's going to continue to lead to a relapse of infection. So that is the current alternative therapy, rather organic for people who like organic or natural homeopathic medications. But that is the current idea right now for how to treat these infections. All right. What we're going to do is we're going to move into talking about measuring bacterial growth again and measuring the effects of antibiotics. So, I'm going to give you one more so you guys can pass one down to Con as well. Now, not only in the lab, but we've covered in class as well as we've talked about the effects of just chemical inhibition of growth and talking about measuring zones of inhibition, performing minimal inhibitory concentration experiments, and we're going to take a look at some data on these types of experiments today and again on Wednesday as well, as we start to talk about methods by which bacteria resist antibiotics. But there are three major ways. One is broth, or the disc diffusion / Kirby-Bauer method, and that's what we have on the worksheet here. Second is a modification of that, known as an E-test. The nice thing about E-tests is this will identify for you actually the MIC of the antibiotic as well. So the design is the same, in which you paint your bacteria onto an agar plate. You then put one of these strips on there. And these strips each contain a different antibiotic, and the numbers here represent concentration. So this one is tobramycin. I think the bacteria is Staph epi or Staph aureus, if I'm not mistaken. And the MIC we would say is 0.064 milligrams per milliliter, or micrograms per milliliter, sorry. That is the concentration at which growth begins to touch at that disc again. So we call that our minimal inhibitory concentration. And then obviously the third way we can do this is by a broth dilution assay to determine concentrations at which antibiotics are most effective and concentrations at which they are ineffective. And again, for these types of experiments, we're looking for the lowest concentration that inhibits the growth of the microorganism. Now, this is different than the MBC, or minimal bactericidal concentration. The minimal bactericidal concentration is the minimal amount needed to kill the microbe. You can't necessarily determine that from this, so you have to perform a second experiment as a follow-up. So for example, this is ethambutol here. Let's assume that this is 16, 8, 4, 2, and 1 micrograms per mil of antibiotic. If it's ethambutol, ethambutol blocks what? Take a look at your nifty charts. What's ethambutol block? What was that?
RNA translation.
Not RNA translation. Mycolic acid synthesis. Mycolic acid would be found in Mycobacterium. So these are all tests performed on Mycobacterium tuberculosis. Okay. So ethambutol here, we would say our MIC is what? Again, 16, 8, 4, 2, and 1. 4 is the minimal inhibitory concentration. And then to identify the MBC, it's going to be at one of those concentrations at or above the MIC. So what we would want to do in this experiment would be to take these last three concentrations, or those highest three, the 16, 8, and 4. We want to just take a little bit of liquid out of each of those. And hopefully that liquid contains some bacterial cells in it, whether they're alive or dead. We take that, we transfer them into 3 new tubes. With nothing but nutrient-rich media, something like LB. Okay. And if the bacteria grows in the absence of antibiotic, that tells us that concentration is not bactericidal. If it's bactericidal, we should observe no growth after subculturing in nutrient-rich media. So if we did this experiment here, we saw growth still occurring with 4 micrograms per mil, but no growth occurring with 8 micrograms per mil in the subculture. We would assume 8 micrograms per mil is our minimal bactericidal concentration. Again, the concentration that it takes to kill the microorganism, not just inhibit its growth. Here's one more practice problem. So take a look at this one real quick. Identify the pluses and minuses represent growth is occurring or growth is not occurring. Identify the MIC and the MBC. So our MIC in this example is what? And again, it's the lowest concentration that fully inhibits the growth of the microorganism. 100 micrograms per mil is our MIC. So that's our MIC. What's our MBC? 200. Lowest concentration that inhibits, where we see no growth in that subculture. And again, that subculture has no antibiotic added to it. So at this point, you're just trying to determine, is the organism that was in one of these two cultures, are they still viable? Are they able to grow at all now when the antibiotic is removed? Okay. So that's how we would perform those experiments. Any questions on these different techniques? Before we go ahead and we spend the rest of the time working on this worksheet together, I just want to see if there are any questions on how to perform these measurements, the interpretation of the measurements, or anything like that. Ellie?
Are you going to post a key for the...
I was, and I completely forgot about it. So I will, if I don't have it done by lab tomorrow, remind me again at lab tomorrow. But I will have it done for you guys, so make sure to put that up. There's a colored pencil up here, so I'll write it down on something before I leave to make sure I note it. Other questions? Okay. So for this worksheet, pull this up on my computer real quick. What I'm going to have you do is work in groups of 2 or 3. And so these are some results from Klebsiella pneumoniae, a clinical isolate of Klebsiella pneumoniae, and testing. This would be, again, one of those disc diffusion assays, testing the antibiotic sensitivity or resistance of this particular strain to ampicillin, colistin, imipenem, what is CTX again? Cefatoxime, amoxicillin and clavulanic acid, which the trade name for this one is Augmentin. And then the last one on there is estrionem, or is that the same as Z-Pak? I can't remember if it is or not. I think it's different. But the case study here is basically a patient comes into the clinic complaining of pain during urination. Culturing of the urine indicates the presence of a gram-negative bacterium. Metabolic testing reveals this organism as Klebsiella pneumoniae. Patient is given ampicillin to treat the infection, but the patient returns 3 days later with intensified symptoms. Antibiotic susceptibility test, or disc diffusion assay, is requested to determine the best course of treatment. What I've given you here, and part of this you'll have to do later because again I forgot to bring any sort of ruler, is measure the zones of inhibition and determine if the organism is resistant, sensitive, or intermediate. And actually I'll probably try to measure that on my own as you guys are working on this. But first what I want you to identify is all of these antibiotics except for colistin are all part of a single class of antibiotics that we talked about. What is that class? And draw out a structure to help you identify it, okay? Then I want you to identify colistin, consider it to be broad or narrow. I will give you the susceptibility data. I'm going to fill out that for you in a minute. And then the rest of the time we're going to talk about how do we decide on a best course of treatment. We've already talked about the effect of broad versus narrow spectrum, but that's not the only thing we have to consider. we also have to consider the therapeutic index. How much is somebody taking versus how much of that antibiotic or chemical will cause that individual to become ill from the actual chemical that you're providing them. Okay? And so that's what we're going to do with the last part of this. Why don't we go for, let's say, let's go until 11:35. We'll take a break. We'll try to answer the first couple of questions, and then we'll finish up with the rest of it. All right? So get into groups of 2 or 3 and go at it. So I looked up the data for the Enterobacteriaceae for these different antibiotics, and based on this and based on these zones of inhibition, this particular strain of Klebsiella is resistant to CTX, ATM, and ampicillin, but it's sensitive to the augmentin, the imipenem, and the colistin. Okay. We obviously want to use ones that the organism is sensitive to. The intermediate here can represent kind of like a transition between sensitive and resistant phenotypes, and so sometimes if an organism fits in that intermediate range, you may not choose to use it if there's a better option. Okay, so that's how you can use this data at least to think about how you would use antibiotics. Well, let's go over those first couple of questions and then we'll continue to move forward. So all of the ones on this list of antibiotics, with the exception of colistin, what kind of antibiotic are they? What group do we place them with? Ampicillin is an example of what? Beta-lactam. All of these are beta-lactams. We'll talk a little bit more about clavulanic acid on Wednesday, but all of these, with the exception of colistin, are beta-lactam. And so the common feature of all beta-lactams, again, are going to be this common ring structure. So they've got double bonded O, nitrogen with a hydrogen group, and where these other parts represent carbons and fill in hydrogens to complete the octet. So that's the common beta-lactam structure. Cephalosporins, penicillins, carbapenems, monobactams, all have this common structure. They're all members of this family. And they all work in very much the same way. There's some difference in activity and toxicity between them, but largely they function in inhibiting cell wall synthesis. Okay. What does colistin belong to? Colistin is a member of what of family or group of antibiotics? Polymyxin. Which targets? Outer membrane, gram-negative. So would we consider this broad or narrow? And this is kind of what Emma and I were talking about towards the end. Given how we've talked about it in class, since it's just targeting gram-negative, we would say narrow. You could make an argument for broad as well, seeing as how it can target all gram-negative bacteria. This is kind of considered a last resort drug because, again, one of the things we talked about is polymyxin targeting the outer membrane. Well, we have membranes in our cells as well. And so there is an argument that there's high toxicity associated with these polymyxin antibiotics. This is not necessarily something you would want to take orally unless you had to. And that's why it has been considered a last resort drug. Not because it's the best and the newest, but because the toxicity level can be pretty bad. We now have strains across the world of E. coli that are resistant to colistin, which is kind of scary because those strains we now have no way of treating them at all. All right, so given this basic information here, I've also given you the toxic dose, which is shown below. And these are just based on a per 50 kilogram basis. So we have to take those two numbers then, and we have to determine which antibiotic is going to be best to use. What are the things that we want to consider? We want our antibiotics to, one, we want the organism to be sensitive to them. What else do we want? High toxic dose, also known as low toxicity, right? It takes a lot of it in order to cause disease or to cause side effects in an individual. So out of these three, I'll do that differently because that doesn't look right. That one, the imipidine, and colistin, come to a decision. Which one would you use to treat an individual? That really is question 5 there, but the other thing you should also consider is why did the individual not respond to ampicillin? Okay, so take a look at the data to support that, to support your argument there. Let's go for another 3 minutes, and then we'll finish up our conversation. Let's try to answer questions 4 and 5 if you can. Why did the person remain ill and symptoms only get worse when treated with ampicillin? There's no zone of inhibition, indicating what? The organism is resistant to ampicillin. So that's why it doesn't work. Okay? Obviously, you can't always figure that out when starting somebody on an antibiotic. Which would we use? Of those three that the organism shows sensitivity to, amoxicillin and clavulanic acid, the imipidine, or colistin, which would you choose to use to inhibit the growth of this Klebsiella and to try and cure the patient of their bladder infection or urinary tract infection? One or the other. I don't know which. I don't know my anatomy. What was that? Oh, I thought somebody said something. Sorry.
AMC.
Why do you say AMC?
Because it has a higher sensitive zone and a lower toxicity dose.
Okay, so the organism is sensitive to it, and its toxic dose is 30,000.
Yeah.
Meaning you'd have to take 30,000 milligrams per 50 kilograms in order to cause toxicity. This is a much higher toxic dose than colistin, and we've already described colistin as being a rather toxic antibiotic, right? What about imipidine? So it's at 35,000. Amoxicillin and clavulanic acid is at 30,000.
That has lower sensitivity.
Potentially. So if you were looking at zones of inhibition, you might look at that. The other thing that I've put up, because we haven't painted a full picture here, is we'd actually need to know, and I can't really find this data right now, what's the therapeutic dose? How much are we actually using inside an individual? From this we can only make some broad generalizations. You'd probably choose amoxicillin and clavulanic acid or imipidine because of the fact that these have high toxic doses and the organism is sensitive to both of them. We'd probably need the therapeutic dose to make a real good decision. Another way to think about this is imipidine is oftentimes, it's one of our newer antibiotics. We kind of reserve our newer antibiotics as antibiotics of last resort. And I'm not expecting you to know that by any means, but you might choose the one that's a little bit older, amoxicillin and clavulanic acid, for a couple of reasons. It's cheaper, and we can deal, I say this in quotations, we can deal with resistance to amoxicillin and clavulanic acid. We don't want to have to deal with antibiotic resistance to imipidine since it is a drug of last resort. Okay. So those are the things that we want to consider in thinking about what antibiotic to choose. The TI is really important. We want to know sensitivity or resistance. There are other factors which we are not spending a lot of time talking about in here, like cost, availability, usage of the drug, that are other considerations to take to consider or to think about as well. For Wednesday, the homework assignment's a group voice thread. So what you and your partners are going to do is each group member is going to take one form of antibiotic resistance, and you're just going to do a video chat and summarize that method of antibiotic resistance and give us a specific example. The last thing that I want to point out, we did not cover the last question on here. Anybody notice this funky observation between these? Pay attention to that. It comes up in the mini lecture, and we'll spend a little bit of time talking about it on Wednesday. All right, see you all in lab tomorrow. Have a great rest of the day.